continuing the cell structure chapters. This doesn't really have anything to do with cell structures, but it does have to do with a career in the sciences, which generally involves going to some school beyond your bachelor's degree. In this case, they're talking about just general graduate school. And just to read through, the thing is, there are no official rules or guidelines for grad students taking vacation. You're not really a student, so you don't get breaks. And you're not really an employee, so there's no paid vacation. So what do I get then? Exploited, mostly. There's a reason I put that up there is right now, this is Sunday. No, I'm in here doing this. I was in here last Sunday. Last Monday was a holiday. I was in here working. Last Tuesday was a snow day. I was in here working. But the thing is, I've been kind of conditioned to work that way. Is that I don't see on days and off days. I just see days that I can get some work done. When you're working in the lab, that's very much what you see. So the next thing on our list of, trying to find my pen. Cell structures, membrane-based cell structures, is endoplasmic reticulum. Endo, inside, plasmic, the plasm, reticulum network. So it's been known for a while. It's got that old Latin name. And it's very often associated with the nuclear membrane. So stuff that can get into this gap can get into the endoplasmic reticulum. And it's a network. It's channels. And sometimes dead ends, like alleys. It's a distribution system for the cell. In some cases, it's a straight up distribution system. Stuff goes in there and it just moves along. In some cases, it's like our throughway here. There's a whole bunch of production facilities or processing centers close to the canalway, throughway. And so materials made and put right into the distribution of it. Looking, this is a transmission electron micrograph. So this is a slice stained with osmium tetroxide. This is what they call rough ER, because there's all this stuff I'll talk about embedded in the membrane, and it looks like if you rubbed it, it would be rough. And the things that are just straight up channels are just membrane are called smooth ER. And that's a mitochondrion, which we'll talk about later. Well, we talked about structures that are found in all cells, and we'll come back and talk about these again. Ribosomes, protein production facilities. That's what's stuck in the membrane in rough ER. So it really is like having a factory next to your road, is that you're making proteins and then you're putting them into the channels that's going to take them to various places around the cell. And if you don't need that, you just need a channel, then you've got smooth ER. Smooth ER also can be a production system. Sometimes lipids, especially lipids that are going to be integrated into internal membranes, are made there. When there's calcium ions in the cell, sometimes because calcium can get into being a charged ion, can get into some systems and screw it up, is that, remember I said there are alleys, like there's blind dead end streets? the smooth ER is a place to kind of sequester calcium ions as well. So there's a few other things that it does other than just being a straight up channel. So here's, again, a depiction of it. Now this is, again, electron micrograph, a little on the overstained side. This is the nucleus. Yeah, I don't know if you probably can't see it's labeled there. There's nothing else labeled on here except this is, you can see the ribosomes embedded in the membranes, and you can see the spaces inside the channels. And again, little bits of mitochondrion, they're all over the place. Now, Golgi was an anatomist. His name is associated with a few human anatomical structures, at least they used to be. They're trying to shift away from anatomy named after people to things that are more, descriptive. Like they want the fallopian tubes to be called the oviducts, Cowper's gland to be called the bulbourethral gland, the vas deferens to be the sperm duct, but that's moving from the Latin to English, and they're not having much luck with that anyway because of vasectomies. This again, made of membrane. It's kind of looks like stacks. Here the stacks are shown to be more open. Here's an actual micrograph where the stacks are pretty flat. In classic depictions, it's almost a pyramid where it's a big flat bubble and then a slightly smaller flat bubble and a slightly smaller flat bubble and then a slightly smaller flat bubble. And what's happening is little bubbles, round bubbles, are coming off the ends of each of those stacks and making them shorter and shorter as you go up toward the cell membrane. I also, before I write this down, it's always named after Golgi, but the second word varies. And I'm sure there's somebody that'll tell you this is not true. But so far as I can tell, there is no differentiation. There's no real difference between whether you're looking at a Golgi body or Golgi complex or Golgi apparatus. I'm sure somebody will go, oh no, there's a difference between an apparatus and a complex. But if that's, I've seen people argue about these things for other stuff that's got multiple names. But I have not seen anybody arguing about Golgi. Maybe I just haven't been reading the right sources. But you know they're talking about something because you're looking inside a cell and it's Golgi somethings. I think the book's actually got 3 links to pretty close to identical pages in terms of material, where one page calls it bodies, one page calls it complex, and one page calls it apparatus. And I don't believe you're going to see that they're talking about anything that's different from one another. Oops. Rats. There's a lot of stuff that's made in the cell that's going to be exported to the outside. That is the secretion. And Golgi bodies is where that stuff is assembled. Bits and pieces are carried by endoplasmic reticulum or other means into the Golgi bodies, and whatever is going to be secreted is kind of assembled in the Golgi bodies. Those bubbles I talked about pinching off move up to the membrane. They move up to the membrane, they fuse with the membrane, and what was in the vesicle is now outside of the cell. And those are called secretory vesicles. Those are vesicles that carry secretions out. And so any kind of cell that's producing hormones or pheromones or mucus, cell walls are partially a product of Golgi secretions and some other stuff that's assembled kind of outside the cell. But there's a lot of reasons why cells export materials, and Golgi bodies are, when you're looking at what's called ultrastructure, you're looking at structures using an electron microscope. What's in the cells can give you a huge clue as to what's going on in the cells. If you see a cell that's just chock full of Golgi bodies, you know that that cell is doing secretions. And when you're looking, like I looked inside flatworms, or not, I looked inside roundworms. I did sometimes look inside flatworms. Is that you don't always know what each cell is doing. So the clues you get is what sort of internal structures are you finding, especially in great numbers, and a cell that's full of Golgi bodies doing some sort of secretions. And then you've got a clue, and maybe you can figure out what the secretion is. Okay. So basically, this is just showing the process. Endoplasmic reticulum conducting stuff to the Golgi bodies, the secretory products made, and pinching off on the ends of these flat things as secretory vesicles. That's, you can't really see it because I'm looking at it without being it on a screen, and that's supposed to be the cell membrane there. With why they would do this, it's almost like you've got the membrane, but they've kind of tilted it so that you can see some of the outside of the cell too, but without clearly differentiating it with a nice dark line. This is supposed to show that this stuff is outside the cell, that this is the surface of the cell underneath it. And yeah, there's names for the cavities inside Golgis. Don't worry about that. Next are mitochondria. One is a mitochondrion. And they've got an outer membrane and an inner membrane. Not supposed to be separate membrane-contained structures inside prokaryotes. You know, the problem with prokaryotes is they very rarely read the rules. This is, at least in theory, connected to the outside membrane in a couple of spots. So you can kind of say it isn't completely separate, but in terms of effect, in terms of how it works, it's basically an internal chamber. Why is that important? I'm talking about something inside the cell. These guys have an interesting history in that at some point in the distant past, after aerobic respiration had evolved in the prokaryotes. Not all of them, but a population of prokaryotes does aerobic respiration. And eukaryotes were evolving, larger cells with stuff inside them. And these guys were potential food objects. So they'd get taken into the cells. But you know, if you don't do aerobic respiration, and this thing you pulled into your cell can produce glucose, or I'm sorry, break glucose down and produce a lot of ATP. It's more valuable alive than dead. It's more valuable intact and working than digested with the pieces absorbed. And so these guys were kept in the cells and put to work. And eventually, cells divided, they would make sure that they got distributed to new cells. These guys would divide inside the cells. Eventually, the genetics of what used to be prokaryotes was kind of subdivided, where genes that were particularly used in here were kept in there. But all of the things that both the eukaryotes and the prokaryotes do, where the genetics was better processed in the nucleus, that stuff was exported to the nucleus, to the point where these things are no longer independent, potentially independent prokaryotes. They can't live on their own, is that they're organelles. This is one of those, I've got a great idea. I think something weird's going on here. What the hell are you talking about? Histories. Is that a researcher named Lynn Margulis back in the 1960s, so 50-some years ago or more, she was looking into the evolution of aerobic respiration and trying to compare how it works in eukaryotic cells to how it works in prokaryotic cells. And so she had a good understanding of how it works and the structures of mitochondria in eukaryotic cells. Now she started to look at the prokaryotes. And the first thing she notices the chemistry is virtually identical, is that it's running the same chemistry, but that could be convergent evolution. How many different ways are there to pull carbon molecules apart in glucose and get lots and lots of energy out using oxygen? But then she started to look at the structures, how the prokaryotes were doing this, where in the prokaryotes the different things were happening. And that's when she started to notice that she might just as well have been looking at a mitochondrion, that the structure and distribution of stuff in these aerobic prokaryotes basically matched mitochondria. And she came up with this idea of, we're looking at ancient kidnap victims. Is the things that independent cells that were taken in and put to work billions of years ago, to the point where they're no longer completely recognizable as prokaryotes, but they're recognizable, but they aren't fully prokaryotes anymore. And that was a pretty radical theory. And it was the 1960s and she was a woman. That was a much larger impediment than it is today. Biology is actually, the proportion of women in biology, they outnumber men by quite a bit now. Not at the upper echelons of the real old guys yet, but we guys get out of the way eventually. Because eventually biology is definitely going to be a pursuit primarily of female scientists, as opposed to physics and chemistry, which chemistry's sliding a little bit that way. Physics is, hanging on by its masculine fingernails. Eventually, it'll all become properly diverse. But there was resistance to the idea, undoubtedly because she was female. And so she kept publishing papers. Look at how this is similar. Look at how this is the same. Look at how this works the same. And it was like, it looks, no, we don't really believe it. You know what's in a mitochondrion? A little loop chromosome. As I said, some of the genes that are particularly used just in the mitochondrion are still in there, but they're not in eukaryote DNA, they're in prokaryote DNA. And that was kind of the slam dunk straw that broke the theory camel's back, is that once she was able to demonstrate there were actually prokaryote chromosomes in these things, it was like, okay, Lynn, we'll buy it now. And it's become basically an accepted, although sometimes it's endosymbiosis. There are variants of these words that you'll run into, but the basic word works. Now this looks like mtDNA, but mitochondrial DNA turns out to be an incredibly important thing for evolutionary studies. Right? We're trying to put together family trees, and we're comparing genetics, mutations from one side to another. Well, some changes in the DNA can be connected to recombining DNA from two different sources, from sexual reproduction. And that can muddy the waters when you're trying to compare stretches of DNA between two different groups. These guys divide inside the cells. They don't mix DNA. Also, when you are conceived, the mitochondria that you wind up with were in your mother's egg cells and not your father's sperm, unless something went fairly wrong. The mitochondria from sperm are not supposed to get into the egg cell during fertilization, like anything in biology. It does happen occasionally, but it's a rare occurrence. It was so rare an occurrence that they were using mitochondrial DNA comparisons for decades before they started to run into instances that didn't make any sense. They went, this is paternal mitochondrial DNA. So they use this for evolutionary studies. One other thing I want to add to this is that this can be found to be happening, is that the idea of eukaryotic cells taking in prokaryotic cells and using them rather than digesting them is still going on today. It's, once you start looking for it, it isn't a process that happened a billion years ago and it just kind of never happened again. There are still protozoa that will ingest useful bacteria and use them. In fact, use them in this next thing. I'm just looking to see what else we got here. Chloroplasts. I'll come back. Mitochondria are roughly football shaped, although they can be long and skinny, they can be perfectly round. They come in many, many shapes and sizes. Chloroplasts, remember these when we talked about photosynthesis? These also are incredibly similar to a particular type of photosynthetic prokaryote. Turns out there's a lot of variety in photosynthesis in prokaryotes, but there is one group that matches this. They use the same chlorophyll, they use the same processes, they're built with the same structure. So this also originated as an endosymbiont, and it's got prokaryote DNA in it. These are the instances that you're more likely to see now, where around here paramecia will absorb blue-green algae and use them as a source of glucose as long as there's sunlight. This time of year with ice and snow on top of the ponds, not enough light maybe getting down there, maybe they'll digest them and then pick up some more in the spring. So they haven't become permanently integrated into the cells, but the idea that they would take them in and use them rather than digest them, something that you can find even around here. Unlike mitochondria, the basic structure of chloroplasts has given rise to leucoplasts and chromoplasts, and I'm just trying to. Some of them are used to store starch, right? They take the sugars and make starches. Some of them are used as pockets to process other pigments, carotenoids. So the blank plasts. There's a few varieties of plasts that are more or less involved or connected to photosynthesis. We're not going to worry about the different types, but I wanted to mention because you may run into, a paper that talks about chromoplasts or leucoplasts. Okay, that's it actually for the membrane-based organelles. We'll do the molecular complexes next.